Good morning, everybody. Please get your seats while we continue with the next session. Thank you very much all for being here. Thank you for the organizers to let us discuss this very important topic for all of us. My name is Doti. I am the head of US Medical Affairs for AstraZeneca. I'm a hematologist by training, and I've been heavily involved in the oncology program for AstraZeneca, which, as you probably know, we've been working in the last years to expand our portfolio in earlier lines. And we have this ambition in the company that we want to share with everyone who works in oncology, that we want to eradicate cancer as a cause of death. Now, getting to that bold ambition is impossible without being good and improving our presence in early screening and detection, and of course, risk prevention. And that is the topic for today and for the panel. We want to discuss what are we doing that is working, what should be doing more in order to accelerate the early screening and detections programs across the country and hopefully across the world. So we're going to do these things a little bit differently. So this is going to be a friendly panel. These are the specialists. They are the stars. I'm just here asking questions. So the first question is, I will ask all of you, please introduce yourself, tell us your area of expertise and your passion for this topic.
Thank you. I would like to start off by thanking NFCR as well for this beautiful meeting. So I'm Ajay Goel. I'm from Beckman Research Institute at City of Hope Comprehensive Cancer Center in Los Angeles. So my research is mostly focused on early detection of gastrointestinal cancers. So within that, I was early on trained with a gastroenterologist, Rick Boland. So I started my career on studying colon cancer, but over the years, I began to study all GI cancers, including colon, pancreas, stomach, and others. And a big part of my research for the last 2 decades have been on developing biomarkers, cell-free DNAs, RNA, other biomarkers for early detection of cancers. In the last decade or so, we have spent quite a bit of time on studying extracellular vesicles and tumor-derived exosomes, because we have learned that tumor-derived exosomes may have analytes which we can use for earlier and earlier detection of these cancers. In addition, we also study the basic biology of RNA, DNA, and also trying to understand the role of gut microbiome. So we all know microbiome is very, very important in terms of cancer pathogenesis. So we try to look at the interplay between gut microbiome and cancer etiology, as well as possibly using gut microbiome also as a possible biomarker for early detection of these cancers. And a very small interest given the disparity of these cancers, especially in a population like the US, where we have different ethnic and racial groups. So we also study racial disparity and biomarkers for finding these cancers in different racial and ethnic groups. Thank you.
I'm Samir Hanash at MD Anderson, and I lead a team of some 40 people. And we call ourselves the rodeo team. We're in Texas. And we are the rodeo team because we cover the entire cancer spectrum, from risk assessment to outcome to detecting cancer early and developing oncology therapeutics. Rodeo. And most of our work is based on the application of proteomics. And I cannot believe it's been 20 years since we organized an AACR meeting on cancer proteomics. And in the past 20 years, the technology has improved considerably, not just down to the single cell level, but also the subcellular level. For example, we cover the cancer surfaceome exquisitely, identifying not only quantifying proteins, but looking at post-translational modifications. You've heard about neoantigens being oncogenes with a mutation. Neoantigens cover a much wider spectrum of changes in proteins represented by post-translational modification. But anyhow, with that slide that you see there, it covers the work that we have done related to lung cancer, risk assessment, and early detection. And so, as we all know, lung cancer is the #1 cancer killer in the world. And at the present time, there are indications for screening for lung cancer for smokers who have smoked 20-pack years. But only 5% of those who are eligible are getting screened for lung cancer using low-dose CT. So what we've been working on for a number of years is to identify blood-based biomarkers that indicate a risk for lung cancer, whether you're a heavy-duty smoker, a light smoker, or even a never smoker. Can we find a marker panel in the blood that indicates your risk of lung cancer? And the answer is yes. I can't believe the work started actually back 15 years ago with mouse models of lung cancer. You would turn on the oncogene, and then we would sample the blood, asking at different stages of lung development, what can we find very early on? And so we identified a number of markers very early on during lung cancer tumor development. And the anchor marker for that was an immature form of surfactant B that we all have in our blood. But this is the pro-surfactant B form. So pro-surfactant B became anchor marker for developing a marker panel for lung cancer risk assessment and early detection. So we identified additional markers that ended up with a 4-marker panel to determine your risk of either harboring or developing lung cancer. And we have done extensive validation of this 4-marker panel in over 1 million subject cohorts, including cohorts in the United States and cohorts in Europe and elsewhere, showing that, in fact, it's very effective at determining your risk of lung cancer to trigger screening for lung cancer. And the most recent publication that we have that came out in JCO a few months ago was using the NCI PLCO cohort, prostate, lung, colon, ovarian cohort, showing that those that died of lung cancer in that cohort, had they gotten that test done early on and followed with a low-dose CT, it would have reduced mortality considerably. So we have not only evidence for risk assessment and early detection, but the gold standard for biomarkers, does it impact mortality? And we're able to show that indeed with a test like this, if applied to even a general population, you could reduce mortality due to lung cancer, more than you could reduce mortality with current therapeutics for advanced stage lung cancer. So risk assessment, preventive intervention, and early detection are going to be very important in reducing mortality due to cancer. Not just finding the wonder drug for advanced treatment, which is like holding a tiger by the tail. Let's start at the very beginning with risk assessment. Who is at risk? We go to the doctor, we get our cholesterol checked. Well, why not go to the doctor, get your blood checked for cancer? We say, well, look, it looks like you have a profile that determines your risk for lung, colon, breast, and ovarian. And then that would lead to intervention. What kind of intervention are we talking about? I guarantee you, before the end of this decade, there's going to be cancer vaccines available so that if a woman is at risk for triple-negative breast cancer or breast cancer, here's the vaccine for you. This is how we are going to reduce mortality due to cancer, by intervening very early on, not just waiting until you have advanced stage cancer and figure out what wonderful drug is going to cure you. That's basically a major activity that we have in part of our rodeo team in Texas.
Oh, okay, thanks. Thank you. This is a great discussion. So I'm happy to be here today. I'm a professor of medicine and vice chair of research in the Department of Medicine at Weill Cornell. And since we're talking, I guess, about our past, just briefly, I'm trained as an internist and a clinical geneticist, and so the focus is on cancer predisposition. So certain individuals, certain families are of high risk for cancer, different cancers. So basically, this is what I tell my patients, there's about 120 cancer predisposition genes that we know about, of the 19,000 genes that cause a predisposition to a constellation of different cancers. It's non-random. So during my career, essentially I've been involved or privileged to be involved in identifying or co-identifying, because these were obviously all team efforts, 7 cancer predisposition genes. And then that was very exciting, and it's wonderful to actually see patients who, actually have been, who have mutations in some of the genes I was involved in identifying. But in particular now, pretty much, and I think Sapna next will agree with this, we pretty much know all, or almost all at least, of the cancer predisposition genes. There may be a couple that are lurking out there, but for the most part, this is done. So now the question is sort of how do we best handle people with high predisposition to cancer? And then of course there are also non-genetic, predisposition, reasons. So for example, we're talking about lung cancer. Lung cancer pre-malignancy is a very clear and sort of established, group. Individuals come in, they, for instance, they have a car accident, they have a CT scan, they have nodules that are found, or non-solid, what are sometimes called non-solid nodules, and then it's unclear really sort of how you best handle these, but these do progress to adenocarcinoma. So essentially now, since Sapna and I and others work with people who have all these cancer predisposition mutations, and then also some of these other high-risk conditions, so what do we do about it? So the focus that we tried to take, and we have an NCI Center for RNA cancer vaccines, or immunoprevention vaccines, or actually it's immunoprevention and interception vaccine. So the concept is really to try to use cancer interception, and a concept that we call cancer dome. And so the cancer dome concept essentially is you try to detect as early as possible, and once you detect, you try to intercept with vaccines. So the idea is to try to use cell-free DNA, these other modalities, identify some of the aberrant, let's say proteins, but as you point out, there are other things like surfactant D is actually interesting. I'll have to read about this. And then try to develop their ways now, working with Drew Weissman and others who won the Nobel Prize for COVID vaccines, ways to sort of tweak the composition of RNA vaccines and the lipids and the lipid carriers and the RNA itself actually, and you can do vaccine co-cargo. There are other things you can put in, for example, various cytokines. There was a recent FDA approval for N803, which is sort of a modified form of interleukin 15 that's used in non-muscle invasive bladder cancer. So there are ways that you can actually detect, if you find some of these things on cell-free DNA, you can not just say, oh, this is bad, but and angst out your patients, but that you have the potential actually to make these more immunogenic and recognized by the immune system. So once again, the idea is intercept, or detect, intercept, eliminate, and because of all the problems we were hearing about, Paul Michel was talking and others, about the myriad, late cancers develop obviously very tumor immunosuppressive environments. We heard that in the first talk from Drew Pardell. Then we heard about some of the tumor resistance mutations that occur, extracellular DNA and chromosomal DNA that make these tumors essentially unresponsive. During Drew's talk, it was interesting and somewhat poignant for me to look at the exciting advances we've seen in immunotherapy, and yet those numbers are not so high, and so we need to do better. And so the way I think we can hopefully try to do better, as Samir pointed out, is to try to use the cancer dome concept and intercept cancers earlier in the paradigm when they're curable. Thanks.
Please introduce yourself.
Hi, everybody. I'm Sapna Singhal. My pleasure also to be here and see so many friends. I don't think we knew who else was going to be on the panel till we got on our first conference call and then realized we all know each other, and have intersected over the years. So I am trained as a gastroenterologist, and I want to go back to what Sam mentioned, which is an analogy that I use all the time, is we would never wait for somebody to have a heart attack to check their blood pressure and their cholesterol. And yet that's what we do for cancer. You know, I think we wait, our approach to cancer is very reactive rather than being proactive. And this has been a fundamental issue in cancer care, you know, for the longest time. And I think sort of gladly, I think we've been changing, largely, I think, first precipitated by our discoveries in hereditary cancer. So I changed my field of interest to be gastroenterology because of an interest in prevention, but found myself, as I was looking for a postdoc, in a room with Richard Kolodner, Judy Garber, and Fred Lee, and these were sort of the fathers of, you know, fathers and mothers of hereditary cancer genetics. Richard had just published one of the first papers describing the first mismatch repair gene. There were parallel papers from Bert Rogelstein's lab from Hopkins. Judy Garber really was working on hereditary breast cancer syndromes, and Fred Lee had described the Lee-Fermani syndrome. And they said, we just published this paper in mismatch repair genes, would you like to work on the GI families? And that's how my work started, and really focused on Lynch syndrome, which is now a very well-defined syndrome. And it's really remarkable how that genetic discovery changed the clinical management of these patients. You know, before it was a mishmash of family history characteristics and, you know, personal age of cancer. Nobody could keep it straight. Once we had a genetic diagnosis, we could actually have targeted approaches to identification, screening, prevention. And it's been a remarkable story over the past 25 years, you know, of seeing this field evolve, where we're actually able to offer screening, surveillance, earlier screening, more intensive screening. We've learned about the benefits of aspirin, chemoprevention, that actually can decrease mortality and incidence in cancer dramatically in patients with Lynch syndrome. And now we have immunotherapy, which can really change the paradigm for treatment of Lynch syndrome. And that's all from the discovery of a basic, you know, the biology of the disease. And so what we've done now is, you know, I mean, starting in Lynch syndrome, but expanded that to hereditary pancreatic cancer. And within the Harvard Cancer Center in Dana-Farber, over the past 5 years, there's been a strategic decision to really focus on early detection and interception. So you notice none of us are using the word prevention that much. Prevention has sort of got a bad name in the sort of lay public. They think of the word as being judgmental and saying you should just exercise more and eat better. You know, so interception is the idea that we're going to actually intercept the progression from in the whole paradigm of cancer development, and it's a term that's being used more and more in the community. So at Dana-Farber and Harvard, what we've done is, within our research programs and our clinical programs, we are embedding identification of at-risk populations in every disease center program. So the GI Cancer Center is now also going to evaluate people with cystic neoplasms of the pancreas to see, can we define which of these people are going to develop cancer? Can we intercept before they develop pancreatic cancer? Can we bring AI technologies to pick up IPMNs and to actually predict who's going to progress versus who doesn't? So we're at a really, really sort of critical time in this, and it's wonderful to see the biotech companies and the diagnostics companies really take this on, because I think it's driven what we're going to be able to do now in the next 15, 20 years to change how we manage care for patients, to figure out, you know, when do we identify people? Ideally 2025, everybody, I don't know if Fumi's here yet, but she's going to be here tonight. She's been a big proponent, and Mary Claire King, of genetic testing early. We're still not doing that. And one of the big things that we've done in my research group is to develop risk assessment tools that we really think should be part of standard care, embedded in electronic medical records, and part of making the patient's care really streamlined and systematic to think about risk assessment across the continuum. So I'll stop there.
Thank you very much. I really love your analogy about the heart condition, because if you see the slide down on the cancer continuum, right, with all the components that you said, and it's superimposed this slide with heart disease is exactly the same components.
Yeah.
Right? So every single thing that you see here is basically the same thing that we can do to reduce mortality in cardiovascular risk reduction. And actually, when you see the data from the US in the last 20 years, the first interventions, the risk assessment, the preventive in cardiovascular has reduced mortality more than 40%. And although we have done a lot of great things in cancer, that does not translate in the same way yet, right? And one of the reasons potentially is you see the amount of money that we spend on research, there's a very skewed amount of money in the latest part of this continuum and not that many in the initial ones. But we know that we will want to eradicate cancer as a cause of death. This needs to shift into the early parts because, as you say, we don't need to wait until cancer starts. We need to prevent it or intercept it in its way to become cancer. And that has a very important component of science. There's also a very important component of education. Because the other thing that when you see this, we're not very good at yet is disparity. Because we can see, for example, in screening programs in cervical cancers being in the US as good as 80 or 85%. However, lung cancer, the average in the country is 7%, with some states being at 1% of patients at risk being screened. And when you dissect those numbers into not only patients at risk, but ethnicity, state, income, and access to health insurance, a lot of the things that we're discussing here could have an impact in some proportion of those patients. But once we unlock that, how can we expand this to all patients at risk and not just the ones that have more access to this? So let's focus a little bit on this continuum. And we're going to talk about the first three things: the risk assessment, preventive intervention, and early detection. So open question for the whole panel. We covered this a little bit. There's a lot of work that has already been done in risk assessment. And there's some things that we can do more. What are those things? What are the next 10 years of risk assessment that we can do?
Well, how do we screen right now?
Everyone looks at you, so you have to answer now.
No, yeah.
How do we screen right now for cancer? Only a few cancers do we screen for. And the indication for screening is based on age. And now, I mean, in the Wall Street Journal, New York Times, what have you, an epidemic of cancer among the young, whether it's breast cancer or colorectal cancer, they're not eligible for screening. Now, why can't we come up with models for risk assessment other than just age and be able to determine who is at risk for what cancer? There have been quite a few papers actually published about looking at millions of electronic health records and coming up with a very good performance risk assessment for a number, including pancreatic cancer. And so we should not be just limiting who gets screened, who doesn't get screened based on age. Your risk profile should determine whether you should be screened for this cancer or that cancer. So your family history, your subject characteristics, and your biomarker panel, or maybe genomic profile, will be the major determinant for whether you should be screened for this cancer or that cancer, and intervene to prevent the cancer in the first place.
So if I would translate that into something for the general public, can we have in 10 years that panel in your annual checkup? Can we have that so the same day that you test for your hemoglobin, your glycosylated hemoglobin, your blood pressure? Is there hope that we will have that in the next 10 years?
Well, I think we have to look at cancer mortality and try to deal with what exactly are people dying of. And there are a few cancers that represents more than 50% of cancer mortality. Let's do a very good job with those cancers and then expand. Let's learn to walk before we start running.
Awesome.
And so let's deal with the common cancers first and then expand to 50 cancers or what have you. That's kind of the concept that we are following at the present time. So we're working on developing markers for lung, colon, breast, ovarian, pancreas, and gastric cancer, the top cancer killers for now.
Also, and that goes to the next topic. So of course, even if we're extremely successful in risk assessment, we're not going to be able to risk assess every single patient in every single tumor type. So the next frontier is for those we cannot risk assess first, how can we do a preventive intervention, right? Not all of them will be preventive, but it's the next step in order to improve the care early in this game. Right? So,
Can I make a comment about
awesome.
the risk assessment part? I do think that we still have a lot more to do to implement what we already know. I know Scott Lippman talked about one of the big challenges of the genomics, precision genomics, is how are we going to get clinicians to actually implement this in general practice? So we've developed these risk assessment models for inherited predisposition called the PREM models. And we just got funded to actually put PREM within the EMR of a Kaiser group in the Kaiser Mid-Atlantic, actually here, near close by to here, Denver Health and Logan Health. And they all serve very underserved populations. And the idea is that everybody will get that tool sent to them before their doctor's appointment. They'll fill out the survey, they'll get their risk prediction, and they will have the option to schedule their genetic testing before they even see the doctor, and then get called with their genetic test results. So I think that's the kind of wave of the future that we need to have, and there's no reason why we shouldn't be screening people for their smoking history and then setting them up for their CT scan, right? One of the biggest problems we have right now, we know CT scanning works for lung cancer screening, but only a minority of the population is actually getting it. So implementing the tools, I think, of what we already know is a huge, I think we can surely do that in the next few years.
Absolutely. So let's go back to the preventive intervention too.
Yeah.
Same question. We've improved our prevention a lot. There's a lot more to be done. What is the next 10 years in this area look like? Anyone can take it. Steve.
Well, so I don't know about 10 years, but you know, I think the hope is that once again is that we can sort of then use the early detection information, and so obviously it depends on what sort of screen. I just want to say that I think Safin and I and others are all really excited actually about the potential for using some of these machine learning tools, specifically being able to communicate the educational component to patients. To be able to sort of, you know, to be able to make it easy for people. Part of the challenge we have with you know, trying to get to interception is to meet the patients in a place where they want to be, which is often on their phones actually, or their computers, in their house. And then to be able to bring them in, or to piggyback it onto a blood test. But once again, once we sort of identify the sort of information, at least, you know, from mutational data, you know, the hope is that we can intervene. So I think it's actually a very exciting time. You know, we see in the post-COVID era, the power of RNA vaccines. You know, an incredible story. I mean, the most effective vaccine ever in history. You know, made in 1 year or 2 years. You know, and so for cancer, we can do this. We can do the same thing. So there's a lot of people doing a lot of sort of exciting work, but the point is to be able to really try to, be able to intervene early with this information, and then the idea is to train the immune system and to take some of the benefits we heard earlier, and introduce it earlier in the paradigm. For some diseases, there'll be more of, for some of the things that Safin and I studied, like these hereditary cancer predisposition syndromes, like Lynch syndrome. You know, there's the potential for off-the-shelf vaccines. Cost is an important consideration, and in like our discussions with some of the biotech companies, and for a lung cancer, pre-malignancy vaccine too. There's they say, we get very strong pushback. It has to be. We want to have many people as possible who can essentially potentially receive the vaccine, and in the simplest, the fewest visits. So we have to kind of reach that. So for some populations, like Lynch syndrome or constitutional mismatch repair deficiency, there's a potential for these sort of lower cost off-the-shelf things. I think these will be in the vanguard. Then going into, the incredible heterogeneity of cancer that we see, the different types and all, there we're talking probably more something that's called the paradigm that like Moderna and biotech have been doing in what's sometimes called secondary prevention. That is, in the adjuvant setting, patient has, say a stage 1 colon cancer is detected, surgically resected, and then they're monitored using for minimal residual disease, with biopsies. And then for example, there are like the Terra and others have, panels that are tumor specific. So prevention for interception, for most cancers beyond some of these things like Lynch syndrome, where the mutation rates are so high and you have the potential for, you have shared mutations, you have the potential for an off-the-shelf vaccine, you're probably looking at something like a tumor individualized approach, based on the cell-free DNA and based on the genetic characteristics of the patient, HLA, and then which is class 1 MHC and other things to be able to integrate into what you would use to vaccinate people.
You want to comment too?
No, I'll talk about the early detection, or we bring it up next.
Go for it.
Yeah, so I think when it comes to early detection, I think we have come a long way in terms of detecting many of these cancers. But I think when we look at individual cancers, we still, just like you said, we haven't done a good job. Colorectal cancer, very, very common cancer. We know that it is preventable if you can find it early, right? So even now, even the data from this year, 50% of the population in the US does not get screened by colonoscopy, who are screening eligible, but they don't get screens. So I think there are multiple reasons. One, access to healthcare, people's personal beliefs that they need or don't need, and then the cost. And I think in terms of colon cancer, the best test we see, or one of the best tests we see on TV screens, Cologuard. So I think that's a wonderful test, but still I think the compliance and participation is still not as high as we would like to see. So if you look at the data even now, I think most of the early detection tests, although we brand them and say these are early detection tests, but I think if you look at the data carefully, most of these tests perform well still in the advanced stages, not in the early stages. So if you look at the Cologuard data, even from 3 months ago, published in New England Journal, so this test still has, very poor sensitivity for advanced polyps, advanced adenomas, or stage 1 cancers. And I believe those are the true lesions we should be focused when we talk about early detection, not a patient who already has a stage 2, 3, or 4 cancer. So we have done quite a bit of work on this. We are actually working very, very closely with Cologuard. So we have a test based on cell-free DNA and RNAs, which has almost 90% sensitivity and specificity for finding patients with advanced polyps. So I think those things are going to change. Similarly, we have a test for early detection of pancreas cancer, which we are almost about to be published now. So I think the focus for early detection, we have to really mean the word early, means earlier and earlier precancerous lesions of stage 1 disease, not later stages. And I think once we can do it, if the test is affordable, it's a blood test, I think we can make a bigger difference compared to looking at some of these tests which really don't work in the earlier stages.
Maybe I'll just say very quickly, I think an important aspect of engaging the patients and once again thinking from their perspective of what they want is having sort of be very clear or that there are potential interventions. So it's not just information. This has come up through the genetic testing paradigm, in terms of cancer predisposition, but that you're not just going to say that there's nothing you can do about it. Be anxious, right? But that specifically there are ways that we can intervene, hopefully with cancer interception, or if not then, at least with intensive surveillance. For instance, for GI malignancies, we do more frequent colonoscopy, and other things.
Yeah, for the early detection tests that are available out there, like obviously the GRAIL test, the performance has not been optimal for several reasons. One is you're going after 50 cancers to begin with, number one. Number two, you're applying it to a general population. And if you're doing that, then you better have super, super, super high specificity. And there's always a balance between sensitivity and specificity. You increase the specificity, sensitivity goes down, and it's not picking up early stage. On the other hand, if you were to identify subjects at risk, then you can afford to lower the specificity and be able to detect the early stage. And so that's where risk assessment becomes very, very important.
I think this is a big tension between the companies that are developing the early detection tests, the multi-cancer detection tests, versus some of the scientists who are studying early detection sort of at the more granular level. But you agree that, I think we all agree that MCED tests, which are multi-cancer detection tests, have tremendous promise, right? They're taking all the technological advancements with the hope of picking up, doing cancer screening for multiple cancers simultaneously. So a patient could go, I would say it's the Theranos story done well, you know, if we could actually have a test that does that, that would be tremendous. But the companies that are sort of developing these tests really want a test for all comers. But as Sam points out, the more sort of parsimonious way to do this is to do risk assessment and then combine that test with the at-risk population. But I think that could also be a paradigm shifting change in the next 5 years. You know, we're going to get the results of these large trials that are ongoing right now with multi-cancer detection. And patients are probably much more likely to do a blood test than to do a colonoscopy. You know, that's just the fact of the matter. And so I think we're going to have a lot more information in the next several years.
So if we see this today,
Yeah.
according to what you're saying, we need definitely to have an integrated approach. It's not just focus on one and not the other. It's an integrated approach to make sure what is the right sequencing for the right tumor type. And it also will require education, right? Because all the science in the world cannot mobilize patients to actually follow up and probably nurse navigation, something to help. I'll give you a small example from something that we're doing in AZ. We have a collaboration with the First Ladies in St. Louis, Missouri, and we sponsor two screening days for prevalent disease in their churches. Right? It includes hemoglobin A1C, includes respiratory screening, smoking cessation learning, and we included PSA. So last year, we were able to test more than 1,000 patients at risk for PSA. We identified more than, I think it was 36 or 38 patients who needed follow-up. You know how many of those patients actually got a follow-up? 2, right? Because we were, and this is our mea culpa, we're focused on mobilizing for the screening, but the most important part is to help that patient get proper care. So it's not only identifying the risk, but educate the patient what to do with that, and even the healthcare system to what to do with that, right? So you want to do?
Yeah. There's been a bill before Congress about MSAT, and the Congressional Budget Office did some analysis of some sort and decided that this is a no-go. It's way too expensive. And so we couldn't figure out what analysis did they do. So we went to meet with the CBO people and asked them, like, well, how did you do your analysis? They said, well, we based it on that one test available out there for $1,000 applied to the general population, and the country will go bankrupt with something like this. And we told them, well, this is not necessarily the only way that you could do some sort of MSAT. It could be a cheaper test, number one, and if you apply it to people at risk, then that could be very beneficial and cost effective. They said, well, we did not know that. CBO did not know that they just came up with a recommendation. They said, please send us kind of like some analysis. And so we have modelers. Some of those worked already with the U.S. Preventive Task Force on recommendation for screening. And we came up with a very cost-effective model for an MSAT test. And so there are ways that you could do it that would not bankrupt the country.
Absolutely. So I would like now to open questions from the audience and make use of this extraordinary panel. Yeah, I don't want to be the one selecting them, so I'll let the people run with the microphone.
Thank you all for your amazing insights. I have a question for you. How do you think the latest FDA guidance about laboratory-developed tests might affect this continuum, especially with the risk assessment and early detection?
That's a great question. I can take the first step, and then we can. I think it's, you know, it's a good thing, honestly, that I think we need more oversight over these tests, but it's also going to put up a lot of barriers for the development of the test. It certainly makes the path more complex. And I think one of the big drivers or the big barriers of early detection has been that there's not been that much investment, right? So it's going to be even a bigger investment that's necessary. So it's good, but it makes me nervous a little bit.
I think you said it all. I think it's going to be a balance between how do we move forward in terms of utilizing the tons and tons of assays developed in individual labs. How do we select the best ones? How do we move them forward? So I think this is something we're all going to learn together.
So Dr. Doti and everyone, this has been a fabulous session, something that we're all thinking about more and more, obviously needs a lot of attention, and ACR is focused on that very much lately, especially. I wanted to congratulate Dr. Doti for what's happening in AstraZeneca, because what we're hearing is that pharma is not interested in this area, and I'm wondering how we can encourage big pharma to invest in early detection. Obviously, smaller companies, Grail, et cetera, have been involved, but when one looks at how to raise money for this area, it becomes very difficult if we can't get the attention of pharma. So I wonder if you could comment on that.
I cannot speak for other companies, but what I can tell you is that we started a journey in AZ in 2012 when we changed our strategy to be more focused on oncology. And we started our journey by entering into the metastatic space in lung cancer and in ovarian cancer. And it was clear to us that the only way to be successful as part of the stakeholders in cancer care was moving into earlier lines. So we moved all our assets into earlier lines, and you see consistently in every area that we work, we have trials going earlier and earlier and earlier. In lung cancer, we move from metastatic to the unresectable, and then we go to adjuvant, perioperative, sometimes more effective than others. But of course, when you see that from a continuum of a company, not investing in early detection and prevention is leaving your work in half. And we even asked internally to our CEO, so what if we're successful and we actually eliminate cancer? He said, so be it. This is our objective as a company, is to transform care. And transform care is not just bring better treatment to a subpopulation of this, but actually to all patients, right? And we know that not every risk assessment or every early intervention will be 100% perfect. So we still work in every single area to transform care. This is very personal, but I'm a hematologist, but I'm a cancer caregiver. I'm a caregiver to a cancer patient. It's my mother, and I lost my father to cancer. So how can I not think about preventing what could have changed my family and for every other family out there? And I think this is the driver, is to have an open conversation on, pharmas are not non-profit organizations, but how we have an integrated conversation on how to transform cancer care from the very beginning, and that starts here.
So this is a question for really anyone on the panel. Certainly we need risk assessment models for evaluating on a population scale. But we have trouble delivering standard of care therapy to disadvantaged communities, whether that be ethnically based, socioeconomic, whatever. How do you plan on helping to implement risk assessment deployment tools where we can't even get standard of care in, which would have the biggest impact in improving health and well-being for those communities?
So I think it's a great point. And I think we all touched upon this in our comments, is that there's lots of knowledge that's not being implemented. And that's one of the biggest things that we do have to fix. That doesn't mean that we don't do other things simultaneously. So implementation of average risk screening, we can certainly do a better job, but there are a lot of efforts in this space in terms of getting out there in the communities, getting out there with the education, and making people I think we have to approach it in the same way that we did with COVID. Remember, at the beginning, nobody was getting vaccinated, underserved populations. When you actually started to reach out to the populations by people from their own communities, the vaccination rates in Blacks and Latinx individuals went up. And in the same way, we need to do that, in our general population. From the risk assessment part, it's even more complicated. And we heard this, Scott Lippin and I were part of a Dream Team award grant to look at pancreatic cancer early detection and prevention. And pancreatic cancer genetic testing is recommended for all comers. We wanted to increase access in our diverse populations. We had very low uptake from Blacks and Latinx populations. And they said, we're not going to do this because in my state, I can't get the surveillance covered, right? My insurance doesn't, Medicaid doesn't cover surveillance. So I think this has to be a multi-pronged approach at not only getting the risk assessment done, but also making sure that people have access to the interventions that they need downstream.
I think reaching out to patients, once again, and thinking sort of like, what do they want? So being able to, once again, use some of these machine learning tools to be able to communicate with people and to bring them into the system. We all know, obviously, that these tools have issues. They have, hallucinations and such. But nonetheless, it's still possible. It's likely. Sorry. The AI tools sometimes say things that aren't true. So there's an error rate, and but this can be improved. So reaching to people, where they want to, and to try to kind of encourage them at least to come in. And then obviously, comparing colonoscopy and liquid biopsy, as we've heard, these kind of things is another way. And is that going to solve the problem? No. But can it make a dent? I think so.
So, this is a brain freeze question, I apologize. But cancer is projected to go up by 2050 to 77% globally, and it bifurcates. In men, it's going up about 87%, and that mortality is going to around 93% in men. That's the projection, the latest projections. So, I spent a lot of years at the NCI, and I can tell you that we have never focused on prevention. I mean, I don't care what we say in this country, we're always focused on treatment, aren't we? Really. And so, how do we change this? This is such a huge policy issue. Everybody on this panel and everybody in this room has to become a pain in the neck or whatever. Because we can't change this until we have some policies that say there are third world countries. There are countries that are going to be decimated by cancer. They don't have the simplest test, Sam, much less, thinking about the proteome. So I think there's a big picture issue that really requires a lot of action. And I don't know how we're going to do it, but I think we have to do it. Does anybody have any good ideas? I mean, I'm serious. This is, 2050 is tomorrow. And we really, if we started today to do the kinds of things you're talking about, I'm not sure how much impact we'd make, unless we have, coming back to Sam's point, almost perfect sensitivity and specificity for the tests that we're creating. Because when you start putting things in a population, a small change in sensitivity makes a big difference. So does anybody have any ideas here for how we get this in front of Congress, how we get it in front of the NIH, et cetera? I think we have to do something different. I said it was a brain freeze question, I apologize. But it's a really important one.
How do we screen for lung cancer at the present time? It's based on smoking history. 20-pack years represents roughly a 1% risk of lung cancer. So the challenge is, for other cancers, can we come up with a profile that represents a 1% risk for that cancer? A 1% risk for pancreatic cancer then would justify wanting to screen for pancreatic cancer. It's not a rare disease in that 1% that are at risk. It's a rare disease in the general population. And so the challenge is, through a risk profile and a biomarker profile, come up with a 1% risk for breast cancer. And for that woman, you better get mammography, or even better yet, get MRI, And if your risk is low, you don't need to get mammography every year. There's a lot of concern right now in other countries about, well, mammography is not cost effective. And so, but if we can identify personalized risk, that's the criteria. Personalized risk for this cancer or that cancer, it will make it much more beneficial, cost effective, and reduce mortality.
Yeah, it's precision prevention.
Precision prevention.
Precision.
Please go ahead.
I have a question. Like, so much we know about what predisposes you to cancer is who you are, which includes your germline beyond the cancer predisposition genes, your immune system, and your age. And for each one of these, we can actually measure. You know, we know a lot about, and you know, they interact with each other. And there were recent studies showing that if you have markers of aging in younger individuals, that means you have higher cancer. So could we envision that everybody, maybe age 20 or 30, get germline testing, get a TCR testing in your blood, and test your aging markers, and then adjust your, you know, like screening and everything after that. So, you know, because up to, especially these younger women and individuals who get these cancers, they are not screened, and they don't have things that we think. And it would be, and it's not only that it determines your cancer risk, but also what type of cancer you get. So there's so much interesting data evolving, and I just don't see that. Why don't we move this into the practice? You know, if we could, it's actually not difficult. We have all the tests available.
I'm just going to make a comment before you make that. 20% of the US population, it's estimated, are on Ozempic. Okay? There's, besides the fact that it's affecting the fast food industry and causing havoc, there is a reduced overall risk to malignancy in that group. Okay? So you're talking about how do you get the system to move forward. But not all of it is associated necessarily with just a reduction in obesity. There are a lot of off-target effects. So bringing together what Dr. Polyak commented on about a better stratification of those individuals and what we now know about Ozempic, people will take it because they want to get skinny. You know, basically, that's how you actually get people on board. They're going to be less willing to go and spend money and have themselves screened. But if they want to, they're vain and they want to look good or they want to get their blood pressure down, they'll go on Ozempic. So I'd like to hear some feedback there. That will bring people to the fore. That'll get people participating.
Yeah, I mean, the effect actually of GLP, it's not just Ozempic, the GLP-1 agonists on the obesity-related cancers, which include, you know, pancreatic cancer, colorectal cancer, liver cancer, and breast cancer, and others, has been dramatic and is exciting. And as you're saying, you're sort of meeting people where they're interested in. So it's like, you know, they're interested in losing and dropping some pounds, and oh, by the way, you're going to reduce your cancer risk. Now, of course, these are associational studies, and so there's some, we have to worry about, say, we don't know about causality as much. We need to more work with some of the, I think some of the, you know, the preclinical models and the more interventional studies in terms of trials specifically looking at this. But that is a great example of sort of something that dovetails a little bit with pharma, maybe, you know, a little bit, and an intervention that's effective and safe. So there actually is likely, and to be honest, I think there's likely good effects. Actually, there's some studies now presented at the ACR conference and others last year in San Diego about the effect of GLP-1 agonists in reducing cancer risk in some of these preclinical models.
I just want to make a comment about the MSET story. Sapna, I thought you were very carefully worded how that's going. And Sam, I personally think this is going to bankrupt the healthcare system. And a lot of people said the train's already left the station, but there's a lot of money, billions of dollars of industry and investors on these MSETs. And if you look, as you said, Sam, if you look at the website, it's to detect 50 different cancers in healthy people over 50. And so I think, I don't know how to do the calculation, there's a lot of money behind this. I don't think it's what March is talking about, where you would do really carefully designed risk modeling and so on. But I think it's a huge problem. I've been very vocal on this for years. And there's actually a National Academies Forum on this topic later this month. Sam may know. I don't know when it is because I'm not going. I made enough enemies on this thing, but those who are interested in the MSED story may want to go. It is a huge issue and has nothing to do with screening risk people.
Please go here. Yes.
I am a dermatologist with a passion for early detection of skin cancer, and it's been my key aim for years. We are now in the 33rd year of our program. Sue knows about this as well. Our survival rate from new skin cancers, including melanoma and basal cell squamous cell, is 100% due to consistent early detection. I'm talking about people who come in, get regular examinations, not people walking off the street with advanced disease. So if anyone who got a new skin cancer, we're able to detect it. We've learned how to pick up the unusual cases with what they look like. We don't use blood markers. We use basically visual knowledge of what happens and how to detect these early. You detect them early, they're all curable. They're all curable. I think the same is probably true in many other types of cancer. You hear of a person who, let's say, suddenly gets a lesion on their back, gets a skin cancer, and they're gone in a year. And they say, oh, this thing suddenly, how aggressive, it suddenly showed up and killed the person. How often do we see a case like that in the program? Answer, none. Doesn't happen. We know it, when we do the examinations, we ask people, bring to our attention anything you thought was abnormal, and they point out some things which are almost always benign. Then we go and find things on their back or other areas they had no idea they had. Patients are not very good at detecting their own skin. And I think there's been too much emphasis on self-examination. Patients don't know what's important. They're not dermatologists. They don't know if they saw something that's a little atypical, they probably wouldn't recognize it. But I think a trained person can do this. We have had a series of what are called physician assistants work with us to do examinations, and their record is also intact, 100% survival. So I think the same thing, once we get the right blood test and it's applied in the right way, I think that we can perhaps approach 100% or maybe even equal that in some other areas where we have the right test done in the right way. And we're doing it, and I think there are certain steps one needs to take, but I think the outlook is wonderful. In many meetings, early prevention is not even discussed. But I think I'm glad the panel and all of you are involved with that, because it is another approach. It's less expensive. You don't have the toxicity, and you don't have cancers coming back later. All of this is avoided if you get consistent early detection. At least in skin cancer, I think it's probably going to be applicable to many, if not all other cancers. Thank you.
You have one more question in the back? Yeah, last question.
So just following on Anna's point, I think it's not only the issue of whether the government's going to put more resources, which is extremely important. I think one thing that's extremely also important in this country is we don't have uptake on the currently available methods. Take HPV vaccine. Some states has less than 30% uptake on this, even prostate screening. So I think this is another thing that has to be addressed, which is maybe even more important because we have available tools and those tools are not being used.
Thank you. So I think our time is up. If I may, first of all, thank you all very much for being in the panel with me. The conclusion, if I may, for this is something important. This is something that we need to do more. There's not one approach. We need to solve the science, we need to solve the access, we need to solve the education to make sure that we reach to every patient. And I don't have a big idea on how to do this, but what I know is that if we go with one approach, so it's only physicians, only payers, or only pharma approach, we will never be successful. Maybe the call to action is put this in the agenda together, everyone who is involved in cancer care, and make sure that this does not wait another 30 years to be a reality. So thank you very much, and enjoy the rest of the meeting.